Thanks very much. It's a real honor to be here with so many distinguished colleagues and hopefully soon to be new friends. And it's really interesting to be the first speaker, not quite knowing what to expect. So hopefully this will meet the needs of everybody. What I want to do is talk to you a little bit about some work that started at Northwestern University with the creation of Biobank that was linked to electronic health records, and then how that's grown with the help of the NHGRI and a consortium called Emerge that I hope most of you know about. And I'll be talking to you a little bit about what we've learned in the prospects of Emerge. So, of course, what drove much of our thinking at Northwestern was the idea that it was clear that there was going to be a revolution that was going to come as a consequence of the human genome. It was well hyped as the human genome affecting the practice of medicine, but to actually think about how do you go those next steps from both using the medical practice to help inform genomics and then going the full cycle to bring it back to how do you actually make improved healthcare as a consequence. And it's been clear that one of the things that we all imagined would happen is that there would be improved ability to diagnose disease susceptibility based on genetic variants that are carried in the genome. We're very far into the process, especially in cancer and a variety of other areas, where disease course can actually be predicted or monitored through the use of genomic information. Sometimes it's used to help treat present optimal treatment strategies, and I'll talk a little bit about that later on in the talk. And then, of course, I think the holy grail is, are there ways that we can think about how to use information in the genome to actually think as a preventative measure for preventing disease? And obviously that would be a huge impact if we were able to do that. One example that I'll use and then come back to a little later in my talk is the idea that not everybody metabolizes drugs in the same way. Nobody in this room will be surprised by that, but it's interesting to know that if you look at hypertensive drugs, maybe about 70% of the people that are prescribed them will actually respond appropriately. And for certain other kinds of drugs, beta-2 blockers used to treat asthma, it's often even worse than that. So the holy grail has been, can we actually predict up front who's going to benefit from a particular medication? And that has several benefits. It avoids side effects in people that are not going to potentially benefit from that. And it also is, I think, probably the first big win for this area of precision or personalized medicine. So what were the key factors to think about defining genetic contributions to disease? Well, I think there are several. First was we needed methods to measure genetic variation in individual human genomes. And when we were thinking about this in 1999 and 2000, it wasn't the case that we had the powerful tools that we have today. Today, where, depending on who you believe is citing the numbers, maybe for $2,000 we can sequence an entire human genome. And that, I think, is going to continue to maybe not get cheaper and cheaper over time, but at least get better and better over time, so we'll have deeper coverage. So we think that piece of it is actually pretty well in hand. The next thing that we really need, if you're going to do both discovery piece and then to think about implementation, is you need large cohorts. You need large numbers of well-phenotyped human genomes that you can then begin to say, these genetic variants correlate with a particular condition or disease that somebody has or a particular therapeutic outcome. Then, of course, what you need is once you've got that information on the discovery side and you've made those associations, you need to start to come up with best practices. How do you actually convert that into care-based decision-making that can be deployed in the healthcare system? And we're still in early days in this process. We're still maybe even in early days in terms of completing some of the associations with disease and with therapeutic outcomes. And then once you get through those basic fundamentals of best practices, then what you need to do is you need to actually deploy decision support in electronic health record systems so that people that are not highly trained genomics and genetics experts can still actually use this information in the practice of medicine. So what I want to do in my time here today is walk you through sort of the evolution that we've gone through at Northwestern and then as part of the eMERGE network to actually do many of these characteristics. So it all began when we started to think about, well, how do we create this cohort or collection of individuals who are well phenotyped? And the idea was that we would create a biobank that was actually linked to electronic health records. So there was my first lesson. So as a PhD non-clinician, I went out and talked to all of my physician friends and said, oh, I'm really excited, we're gonna do this, we're gonna create a biobank linked to electronic health records, and we're gonna use that to phenotype people. And then immediately they like frowned or sneered or laughed at me, and they said, well, you're crazy because the information in electronic health records is not good enough for research purposes. Now, to me, that was really devastating news, not only because they were suggesting that maybe this grand scheme that we had wasn't going to work out so well, but it's also devastating because it sends a really interesting message, and that is, it's not good enough for research, but somehow it's good enough for our healthcare. And that seemed to me a real problem that we needed to think about addressing. So we boldly went ahead and said, we're going to try to figure this out. So in 2002, we launched an Irb Approved Project. It's a completely voluntary process on the part of the participants. They agree to sign a consent, and what they agree to in that consent is to allow collection of biological specimens. Primarily, initially, it was just a blood sample that we could extract DNA from, and then link that information to the information that was in their electronic health records. And we were really fortunate because at Northwestern was one of the first early adopters of electronic health record system, and when we started, we already had about 5 to 7 years worth of data in our electronic health record system. We also were able to use a very interesting feature which has been extremely powerful, was the ability to recontact. And about 90% of our participants agreed to check a box that says, you know, it's okay for you to recontact me to ask whether I might want to participate in a follow-up project. And that has been exceptionally powerful. So we built this as a resource to facilitate genomic research of the sort that I've talked about. And one of the interesting things is that we required all investigators that use these samples to agree to redeposit any genomic or genetic information that they identified back into the database with embargoes so that they could have time to publish their papers. But that has also turned out to be a very enriching process over time because the data set has grown richer and richer. As I said, the informed consent allows a sample of DNA to be collected. It allows access to electronic health records data. And by this, I mean really anything related to the healthcare system, not just the EPIC or Cerner or whatever electronic health records data, but all the other ancillary systems that surround healthcare in an electronic fashion. For one of the few times in my life, I happened to stumble on by accident the idea that instead of talking about using it for a particular assay, we were just going to ask for permission to use it for any type of genetic variation measurement. And of course, starting in 2002, that's turned out to be very helpful because we're not measuring genetic variation anywhere near the way we did in 2002. And so we can continue to use this for information. And the other thing that is we were very clear about is that we were going to share this data very broadly. And the consent was very clear about the data sharing. So that allowed us to easily deposit in national databases. It allowed us to easily even share information with for-profit companies because we believe that at the end of the day, that's the only way some of this important information is going to get commercialized into the healthcare system. So the way the system works, you can see it here, is a recruiter meets a participant in the context of them receiving their health care. In some cases, this is a prearranged meeting. In some cases, it's what I call a cold call. The recruiter will meet them while they're sitting in the waiting room. And if they agree to participate, then they sign the consent form, and then they fill out a questionnaire that gives us a fair amount of demographic information, gives us some self-reported history, health histories. By the way, it's very interesting to compare the self-reported health histories with what the electronic records say their health histories are. And it gives us access to, as I said, all of the systems. We did want to promise as much confidentiality and privacy as we could, so we've separated the system, as you can see here, so that all of the data on identifiable pieces of information, all the identifiers are separately held in a separate, cryptographically separate database that can be connected only with appropriate consent from that particular individual. And that's turned out to be pretty powerful in terms of our ability to use the data in a de-identified way, but still have pretty rich information. So where is NewGene today? Well, we're at about 11,000 participants enrolled and consented. Not the biggest such biobank, but it's one of the biggest that's fully consented, and that's a key element for us. As you can see, it's about 42% male and 58% female. That skew primarily reflects usage of the healthcare system. Many of us who happen to be of the male persuasion go because we're nagged by our partners to go see the physicians, whereas women are much better about taking their own initiative in healthcare. We have an age distribution actually that includes both some pediatric samples and people well over 100. And we're very proud of the fact that in Chicago we have a pretty diverse ethnic population, and our diversity in our biobank matches that in the Chicagoland area. One of the other interesting features is that our participation rate is about 28% based on these sort of random cold call encounters in the waiting room. But if we can just get the physician to mention that there's this interesting study going on, participation rate goes up to an overall rate of about 50+%, and actually in many clinics that goes up as high as 85 or 90%. So it's really good to have the physicians on your side. And as I said, something like 92% of the people agree to be recontacted. I've already mentioned the questionnaire. They complete it once. But it's exceptionally useful for us because it's one of the few ways that we can capture some environmental information, and I'll show you one more in a few minutes. We can access both electronic records and billing data, and the advantage of electronic health records is by its own nature, it's a longitudinal record. As the participant stays in the system longer and longer, obviously their electronic health records data gets more and more robust. And just to give you a sense of that, you can see here, and this was from numbers when we had about 10,000 participants in our biobank. If you looked at the diagnoses that had been given to our participants, something like 34% of our participants had an essential hypertension diagnosis. Something like 16% had overweight or obesity monitoring. It's actually scary, some of the BMIs that are in our collection. 14% have diabetes, and those of you who know the US statistics at least, this matches pretty well what the population demographics are, saying that we're getting a pretty decent cross-section of disease burden in our cohort. One of the other things that's exceptionally rich about electronic health records is you tend to have repeated measures. So you don't have to worry about, was that one measurement you got, exceptionally precise, because we actually can look at trends over time and we can look at averages. Again, when we had about 10,000 people in the bank, you can see we had about 8,500 people that had glucose measurements. And for many of them, those were repeated measurements because we had about 265,000 measurements for glucose in our electronic health record system. And what that allowed us to do is actually really look across trends, and by doing averaging or taking medians, we could believe that we had actually pretty high quality data. And this ability to have multiple repeated measures turns out to be very important because it does allow you to follow trends over time. And then the other thing is, because we had permission to look at where they lived in terms of census, it allowed us to start to think about geographic variation. And one thing I won't have time to talk about today is we've used this approach to start to talk about how can we measure environmental impacts by linking up census data and where we know people live with Environmental Protection Agency measures of environmental pollutants. We can map them now along major highways in the city of Chicago, and ask what the effects of that are. And we even started a project where you can start to look at where food island deserts are occurring, so where people are far away from strong grocery stores. And of course, that has a strong socioeconomic impact. So you can see we cover pretty broadly Chicagoland area. So this biobank, as I said, was developed to do two things. It was developed to help us do discovery, to make new associations between genetic variants, disease susceptibility, and therapeutic success. And then it was also developed to help us do implementation, whether you call it precision medicine or personalized medicine. The whole idea was, how can we use information in the genome to better inform clinical care? And one of the ways that has grown very nicely in the United States is through something called the Emerge Network. A few people in the audience here are very familiar with that. What you can see is it's a consortium of about 12 individual medical centers across the country. And what they all had in common, like NewGene and like Northwestern, was they had a biobank that was linked to electronic health records data. And so they were able to, all the members of the Emerge Network were able to analyze their data and then share their data across sites, which turns out to be important because, as the title of the session says, size matters. So while 11,000 is nothing to sneeze at, when you can across the entire Emerge Network get 10 or 15 or 20 times that, it really gives you an enormous resource to begin to provide the important data. So the first business for the Emerge Network was to finally decide, yes or no, data and electronic health records is good enough to do research. It was also to figure out, how do we develop and validate electronic phenotyping algorithms? And then, of course, how do we do association studies that discover new phenotypes? So to give you a sense of what Emerge has looked like over time, and Emerge is about to enter a phase 3, but by the end of Emerge 1, we had about 100,000 participants in Emerge 1, with about 18,000 of them with at least GWAS level of data. By the time we entered into phase 2 of Emerge, we were at almost 350,000 participants, and that number now is up to about 55,000 genotyped individuals, and actually that's growing by the day. So to address this first question of, is electronic health records data good enough, we thought we would make an approach that says, if we look at type 2 diabetes in individuals whose ascertainment of their disease is simply based on electronic health records data, can we identify the same genetic variants that have been identified in purpose-built cohorts of type 2 diabetics that were created for genetic studies? So what you can see is that one of the things we learned early on is it was not sufficient to just simply look for a diagnosis code of type 2 diabetes. You would fail if you did that. So hence, some of people's skepticism about electronic health records. But as you can see here, if you actually have an algorithm that includes not only diagnosis codes, but laboratory values and medication information, if you're able to actually use natural language processing to read the physician's notes, turns out you can get a positive predictive value of a type 2 diabetes algorithm that far exceeds 95%. And if you do the genomics on it, what you can see here, and I'll just show one example, probably the most famous type 2 diabetes-associated gene is the TCF7L2. And you can see that across the Emerge network, we had no trouble achieving genome-wide significance for this and a variety of other type 2 genes that have been now associated with type 2 diabetes. So we believe, based on this proof of principle, that at least in the case of type 2 diabetes, we were able to show that you could use information mined from electronic health records to find the same genetic variants you would find if you built a purpose-built cohort. The beauty, of course, of electronic health records is that most of us have more than one disease in our history, and this data can be reused in multiple ways. So I want to show you that across the Emerge network now, initially we set up to look at really 5 or 6 phenotypes: dementia, cataracts, peripheral arterial disease, type 2 diabetes, and cure restoration. at Group Health, University of Washington, Marshfield Clinic, Mayo Clinic, Northwestern, and Vanderbilt. But by sharing the data and sharing these algorithms across the other institutions, we were actually able to expand the statistical power of the entire network. And ended up with, in the case of eMERGE-1 and for the project I'll tell you about in a minute, 17,000 samples that were primarily done on the Illumina 660W chip. So the question that we ask is, can we use this data set to identify new novel variant associations with disease without any additional genotyping? So the idea we pursued was primary hypothyroidism. The most common form is chronic lymphocytic hypothyroidism, also maybe known as Hashimoto's disease to many of you. It's fairly common in females. And as of the time we began this study, which was in about 2011, there had been no published GWAS studies on it. So it seemed like good fertile ground for us. As is always the case, we had to create an electronic algorithm. I won't go through it in detail, but to remind me to say that whenever you have an algorithm for cases, you also need to check all of those same elements in that algorithm to assure you can identify controls that have a high probability of not having the disease. As you can see here, across most of the sites, we were able to get case and control positive predictive values in the 90s. A couple of cases it was a little lower than that, but it was a fairly robust algorithm that worked very well to identify people with this condition. And then we went on to do a genomic analysis, and as you can see, got a very strongly significant hit, which turns out to be in a very interesting gene, which is FOXY1, which is a transcription factor that's important in thyroid development. So not only was this a novel finding, but it was something that was biologically plausible. And we've gone on to study a little bit more about the features of FOXY1 and how it contributes to Hashimoto's disease. But I want to use this as a tipping point to talk about another advantage of having electronic health records information. And that is something that was developed by the eMERGE network, led by the folks at Vanderbilt, and that is the concept of something called a phenome-wide association study. So think of this as a GWAS study stood on its head. So imagine taking a particular genetic variant and then looking across all of phenotypic space and asking the question, what phenotypes are now associated with that genetic variant? Hopefully everybody was able to follow me on that. So what we did basically was took all of, the first pass of this was simply to take all diagnostic codes, combine them together so that they fit into categories of related diagnoses, and then ask for this variant in the FOXY1 gene, what other phenotypes might be associated with it. So you can see the good news is, of course, in this analysis, hypothyroidism shows up. It should. That was how we selected that gene in the first place. But then you can look and see that there are other things that reach genome-wide significance. So this is the Bonferroni correction threshold. So thyroiditis shows up, non-toxic nodular goiter shows up, and a variety of other things show up. So it's a very interesting approach to start to think about now, how do you look at pleiotropy? How do you look at the ability, using this approach, to think about other genetic effects of a particular genetic variant? And so this is a powerful approach I hope everybody has heard about because we think that the PheWAS approach is going to be very powerful going forward. So the conclusion from this was that, first of all, an experiment that reanalyzed data based on information in electronic health records and a completely new phenotype was able to identify a new genetic association. And that this FOXY1 was a likely candidate, but that we were also able to demonstrate using this PheWAS-based approach that you can learn something else about other conditions that genetic variant might contribute to. I promised I would talk a little bit about pharmacogenomics. The eMERGE network as a network has taken on a project that we call the eMERGE PGX project. And the opportunity here is to take a panel of 84 genes that have been identified as important for drug metabolism and to deep sequence all 84 of those genes. And the idea is twofold. A, we will uncover some genetic variants for which there's already pharmacogenomic information. We follow the CPIC guidelines. So if there's a CPIC guideline for a particular genetic variant that suggests how you should either dose or alternative medications that should be used in that case, the goal of this PGX project is to put that information back into the electronic health record and actually see if it makes a difference in terms of the health care of particular individuals. But the second thing that will happen in the consequence of this, in the context of this project, is that we will identify a whole slew of new genetic variants because of this deep sequence information that we did not otherwise know about. So this is a list of genes for which there are well-developed CPIC guidelines. There's actually a few more of them now. And the goal was for as many of these as possible to take and measure these genetic variants and put that information back in the electronic health record. I want to talk a little bit about the structure that we've developed to use this, because I think it's informative. So the way it works is we start with information that comes from the sequence work that we've done. And in most of the sites, that sequence of these 84 genes is being done in non-CLIA or non-clinically certified laboratories. So we're in a mode right now that for some of them, we are also having to validate those in a CLIA laboratory so that we can actually return that results for clinical care. One of the things that we hope is by pursuing this project, we'll actually be able to fairly soon get out of this mode, where we require this CLIA individual certification, because obviously this will work best if you can just do the sequencing and use the information directly from the sequence. So we put all of the genetic variants into what we call an ancillary genomic system. And the idea here is not to clog up the electronic health records ultimately with the entire human genome sequence for every participant, but rather to collect them in some way that they can be used for research purposes, but then to filter that data with some kind of evidence of actionability. So that if there's an actionable variant, that gets sorted through our ancillary genomic system, and then that information gets sorted through a knowledge engine, and where there is a good bit of information or evidence for a particular variant, that variant then gets put directly as a lab value into our electronic health record system, which then is available to physicians both as a lab result, but it also allows us to fire best practice alerts or clinical decision support. I should say one more thing that's a nice feature of this is that information is also instantly and immediately available to the participants through their electronic health records portal. In our case, it's called MyChart. So we've developed in the ancillary genomic system, and this is for those of you that are informatics types that might be interested, is pretty standard entity attribute value model that lets us keep track of all of these variants, and then based on that information, create computed observations. So for the example, a computed observation based on some of this information might be that for clopidogrel metabolism, based on the genetic variant this particular individual has, they're a poor metabolizer. And that information then is what gets presented to the electronic health records data. On the physician side, what you might see, and this shows just some clinical decision support that fires. So this would happen in the case of this particular participant being given a prescription for warfarin. Then this clinical decision support shows up that says, this is what we know about the genotype of this individual, and this is what you should do based on the genotype of this particular individual. And then it goes on to say that, for example, for this participant on clopidogrel, that this patient is there but might be a poor metabolizer. And so you can see that the medication might be ineffective, consider an alternative. And then you can actually click on some of these links. It will take the physician who's facing the patient to some additional information that they can either review in advance of their meeting with the patient, at the time of the meeting with the patient, or subsequent to that. And then we collect, this is a research project, of course, so then we collect whether they actually use that information and what they've done with that information. So just to give you a couple of ideas of preliminary observations, focused on two channel genes, both sodium channel and calcium channel, potassium channel. Of first 2,000 participants, we identified 122 rare alleles in 223 participants. So this is a surprisingly large number, and this is what any of us that are going to be doing large-scale sequencing on people whose information is in electronic health records are going to face. We're going to have a flood of these kinds of variants. There were 42 rare alleles in 63 participants, rare here meaning they were present in less than 0.5% of the population, that were actually designated in at least one data source that does such evaluations as potentially pathogenic. Yet, if you compare across people that had these alleles in our system with people that did not, there was no difference either in terms of arrhythmias or electrocardiographic phenotypes. So it's a very interesting problem of penetrance and how do we start to think about this. At Northwestern, we had one participant that, turns out, when we went back and looked at the electronic health record, actually had an example of long QT that hadn't been diagnosed. And so this was an opportunity then to follow up, and the person's physician has already followed up with them and actually changed the course of therapy based on that. So to conclude then, it's important also that the participants can see this genetic test results, and this gives them an opportunity to see in their patient portal some information, and that even links out to some other resources. So in my short time with you here today, I hope I can leave you with a few conclusions. First of all, that biobanks and electronic health records are increasingly playing a critical role in identifying new associations between genetic variation, disease risk, drug efficacy, and clinical outcomes. Longitudinal mining of electronic health records can be used to provide the most up-to-date phenotype associated with human biospecimens. I didn't have a chance to talk about it, but actually by going through this process, we found that we can improve the quality of data in electronic health records. And I'd be happy to talk to anybody more about that. eMERGE has really proven for sure that the networks of electronic health records biobanks that share samples have the increased statistical power to make new genetic associations and to test new hypotheses, and that methods to store genomic variation in the electronic health record have been developed, and the ability to return that results in conjunction with clinical decision support is an important tool going forward. So I'll stop there. Hope I haven't gone too far over time, and would be answer questions if there's time.
So we have time for 2 or 3 questions, and microphones are in the room. I'll ask you your first one, because I'm still a skeptic from some of the information I see in medical records. I used to run an asthma clinic here up the street, and I know many diagnoses of asthma were not when we actually verified them, in the lab. But I'm actually wondering, what are the improvements that are on the way, or what have happened or on the way in terms of ensuring that we have better quality in the EHRs?
Yeah, thanks. That's a great question. So there are a variety of improvements that I think that are making the health records better and better every day. They include things like the fact that in the United States, at least, people are being measured by their ability to exchange information between different sites, called meaningful use in the United States. That means that people are using more and more standardized terminologies. It means that the information in the record is constrained. One of the things that we found in our records was interesting. If you looked in the electronic health records and looked at the birth weight of babies delivered at Northwestern University, there were a few 1,000-kilo babies. Mm-hmm. It turns out there really were no 1,000-kilo babies. It was just that the wrong unit was entered. Mm-hmm. Once you constrained the field so that they could enter only either kilograms or grams and not both, that fixed that problem. So I think lots of examples like that are going to continue to improve the quality of the data. And I would argue that research uses such as ours, I mean, we were the ones that discovered the 1,000-kilogram babies. And so that we were able to go back to our operational health records folks and say, hey, constrain that field and everything will be okay. And it was, and it's already improved the data.
That's good.
Thanks. You make me very hopeful about medical records. My question is regarding the rare variants that were picked up. Are those available to the participants through their MyChart? And if they are, how much do you worry about the anxiety level that all of this is going to create?
Great question also. So the answer is no. One of the reasons I talked about the ancillary genomic system is that this is a system that sits outside the electronic health records. So that collects all of the variants. And the only ones that make it through into the electronic health record is something where there is sufficient evidence to say that there's a clinically relevant actionability that can be taken based on that association. Now the hope is over time that evidence will grow and more of those variants will be able to flow into the electronic health records data. But at this point we're only putting in things that there's at least some evidence that says it's actionable.
Thank you very much.